everyone, I am Varshanjali. I am here to discuss a few points about restriction enzymes. Actually, restriction enzyme is also called as restriction endonuclease. It is an enzyme produced chiefly by certain bacteria that has the property of cleaving DNA molecules at or near a specific sequences of bases. Here comes the introduction of restriction enzyme. Molecular scissors is the other name of the restriction enzyme because they cleave at or near specific recognition sequences known as restriction sites. Restriction enzymes are mostly found in bacteria. Werner Arber discovered restriction enzyme during 1960s. As we moving to the next slide, we can learn what are restriction enzymes actually. These restriction enzymes are found naturally in prokaryotes. It is an important tool for manipulating DNA and most bacteria use restriction enzymes as a defense against bacteriophages. And next one is mechanism of action. In this process, restriction enzyme calculate and scan the length of the DNA and binds to it, and then it recognizes the specific sequence and makes one cut in each of the sugar phosphate backbone of DNA by phosphodiester bond. Restriction enzyme cut DNA bonds between 3 OH of one nucleotide and 5 phosphate of the next one. Uh, there are two ends of restriction enzymes, blunt end and sticky end, whereas blunt end is called as non-cohesive end. In blunt end, some restriction enzymes cut DNA at opposite base. They leave blunt-ended DNA fragments. These fragments can be joined to any other DNA fragments with blunt ends. Whereas in sticky ends, most restriction enzyme makes staggered cuts. It produces single staggered sticky ends. And next, there are three types of restriction enzymes. They are classified based on their composition, enzyme cofactor requirement, nature of their target sequence, etc. There are type 1, type 2, type 3 restriction enzymes. And in type 1, restriction enzyme cut the DNA far from the recognition sequences. They do not produce discrete restriction fragments, hence they are not much practical value. And whereas in type 2 they cut at specific positions closer to or within the restriction sites. Discrete restriction fragments and gel banding patterns are observed. They are exclusively used for DNA analysis and gene cloning in the laboratories. These are a family of unrelated proteins. They are named after the bacterial species from which they are isolated. And the example is E. coli. It is isolated from bacterial species E. coli. The restriction enzymes generate two different types of cuts, blunt ends, and are produced when they cut the DNA at the center of the recognition sequence, and sticky ends produce an overhang. And type 3 restriction enzymes, it is a group of endonucleases that recognize a non-palindromic sequence comprising two inversely oriented sites, cleave DNA by long-range interaction between sites in both head-to-head and tail-to-tail inverted repeat. And there are wide range of applications in restriction enzymes. These are used in gene cloning and protein expression experiments. Each of these methods depends on the use of agrostyle electrophoresis for separation of the DNA fragments. Restriction enzymes are also used in biotechnology to cut DNA into smaller strands in order to study fragment length differences among individuals. They are used to assist insertion of genes into plasmid vector during gene cloning. Isolation of restriction enzymes are used to manipulate DNA for different scientific applications. And the biological role of restriction enzymes is the lost DNA is protected by methylases, which add methyl groups to